I get my slides up? There we go, deep learning. That's what we're going to be doing. So I don't know how I'm supposed to feel when Andrew says my talks are full of it, but we'll see how this goes anyway. Let's see, there should be some sort of a way for me, maybe down here. Okay. All right, so I'm going to cover sort of in a way that sort of goes through different imaging modalities in a way that kind of introduces you just at a high level to different deep learning approaches. And so I'm trying to lay the groundwork for, you'll see a lot of applications as Andrew mentioned later in the talk, or later in the session here. So our goal is to really use these techniques to find subtle patterns in the data, minimize assumptions. How many people see something in here besides rocks? Anybody? How about now? So there actually is a person in there, and it's just meant to illustrate that what we're hoping we're doing is finding patterns that are actually real, that are in the data, as opposed to finding something to make up stories about. And I think you'll be convinced of that by the end of this session. In speaking of sort of flexible modeling, data-driven approaches, Thomas Edison used it as an interview question. He asked people if they would flavor their soup before they tasted it, and if they said yes, then they wouldn't get hired. His assumptions are innovation killers was his stance. And Leonardo da Vinci actually had some deep learning experience. He said the deeper the feeling or network, the greater the pain. So the basic unit just of deep learning is an artificial neural network, and essentially what we have are inputs that are multiplied by a set of weights and then added up, and then you might have something like a perceptron at the end, which gives you a 1 or a 0 based on the weights, and then you can combine those in lots of different ways to create an artificial neural network. And there's a hidden layer, and an output layer, and an input layer. The number of hidden layers can grow quite a lot. As you probably know, deep learning is very popular these days for a lot of reasons that have hit the press. So for example, it's mastered Go. It can beat the top Go experts now. Deep learning can play video games competitively with others. You can use it to do all sorts of cool things like imitate style, where you sort of combine a picture with a certain style and it creates a stylized photo, and there's a bunch of cool editing applications of it. In terms of milestones, there's been sort of ups and downs throughout the past 50, 60 years and more. This perceptron came about in the 50s. The multi-layered perceptron started here in the 80s, and it kind of got a lot of interest and excitement, and then it didn't work very well, and it subsided. Support vector machines came on, and then here's where sort of the modern deep learning age occurred, again with the same person here, Jeff Hinton. He stuck with it, and they figured out how to train these networks and do pre-training and other kinds of ways of improving the performance. And what really convinced people was this example from AlexNet, where they had a bunch of labeled images, and deep learning methods started to substantially outcompete other existing models. And you can see here where the number of deep learning methods started making an impact around 2012. And since then, it's continued to increase performance and increase the number of models that are used. And that's even old at this point. Deep learning architecture started out with a few hidden layers up to 5 in 98. The ResNet model in 2016 was 56 layers. They tried a 1200 layer model, which didn't work as well because probably due to overfitting. These days, I would say the most useful networks are in the sort of, there are a number of models that are sort of in the hundreds of layers at this point. And it's also transferred to other fields, including neuroimaging now. So, detecting subtle things from images that can predict future outcomes. For example, retina, looking at retina, looking for skin cancer, gene expression. If you look at sort of the impact of deep learning in brain imaging, you can see here it's doubling, more than doubling every year. Just from last year to this year, it's already doubled, if you're just looking at brain imaging and MRI. So how do we do deep learning? At this point, it still is, in many ways, a bit of an art as well as a science, because there are many, many models. This is just a small subset of different ways that you can combine these neural networks to do different tasks. And I'll cover some of these. So starting at sort of a basic model called a restricted Boltzmann machine, and this is really just a shallow layer, but it's using something called Gibbs energy, and it's optimizing in the context of a neural network. And if we use a restricted Boltzmann machine, we can get performance in brain imaging data that's sort of comparable to what we find if we do independent component analysis. We get similar looking networks from resting fMRI data, and compare it to some other models, and you get fairly similar performance. Why that's useful is because restricted Boltzmann machine is sort of the building block for deep models, and so we can then start to stack these things and get to more complex models. So here's an example where we go from a restricted Boltzmann machine at one layer to multiple layers to analyze structural MRI data. And what we find, initially we weren't sure if this was actually going to be useful, but it does appear to help in terms of, for example, classification. So here we have raw data, one layer, 2 layers, and 3 layers, and testing and training data is both shown here. And this is just testing for a difference between schizophrenia patients and healthy controls. Gray matter segmentation images are the input. And you can see it actually improves the separation for both the training and the testing data. And this is true in more complex problems. We're all trying to figure out mental illness and the right way to sort of label those data sets. But if we just use sort of standard DSM-4 labels, you can see here, if we start with, again, depth 1, depth 2, depth 3, you are getting an improved separation between healthy controls, schizoaffective disorder, which are schizophrenia patients with a mood aspect, and then bipolar disorder and chronic schizophrenia. And this, again, is structural MRI data. Another thing that kind of convinced us early on that this was useful is, we used it to sort of classify Huntington's disease from healthy controls. So here we have about 3,500 scans, again, gray matter. But then when and what I've done here is I've plotted just a scatter plot. This is t-distributed stochastic nonlinear embedding. It's really just a 2-dimensional representation of the output. Each dot is an individual data set. But then when we color code it by disease severity, and we didn't tell the model anything about that, you see this nice gradient here from least severe to most severe, and here are the healthy controls. So it sort of learned this information from the data without us telling it about that variable. So a variation on deep learning is deep residual learning, which is really, again, each of these things is a layer, and what we have are these sort of shortcut loops here to try to improve performance so that the error doesn't get lost in the large model and it doesn't get optimized for. And so this has been shown to work quite well in a number of problems. And we, again, have applied this to structural MRI data in the context of Alzheimer's disease, mild cognitive impairment. And here you can see various groups here. We have healthy individuals, stable MCI, so they have cognitive impairment, but they don't get worse over time, progressive MCI, and then Alzheimer's. And so we're getting about 93% separation of controls in Alzheimer's, and we're getting a reasonable separation between stable and progressive MCI of about 80% here. And importantly, and I believe this will show up in multiple of the talks later, opening up the black box, I think, is really important for brain imaging. And so you can do things to try to visualize what's going on in the weights that we fit in a deep learning model. Here is just an example of just looking at the filters for the first layer of a given model, and we can project that back onto the brain and see what is important at that layer. And there's all sorts of ways to do this. In the interest of time, I'll jump over that and just go straight to a nonlinear ICA example. So here, we basically have multiple MLPs, perceptrons, and what we're doing is creating a model where we're optimizing for independence, but allowing for a nonlinear decomposition as opposed to just a linear. And we can show that it does a much better job of finding this is a ground truth, and this is the nonlinear case. And this had a nonlinear, obviously, quadratic relationship embedded within it that linear ICA wasn't able to find well, which you would have expected. And then when we looked at the real data, so the question is, is this something we actually need? And so we look at the real data, and when we remove the linear effect, we actually find several covariant gray matter components that show nonlinear relationships. Others are more linear and probably don't need the nonlinear ICA aspect. But so it does appear that there's actually some informative nonlinearities in the data that we can model with this approach. Another important area is sort of training. How do we train these models? And how do we get enough data to train these models? And so one way that's used a lot is called data aggregation, which essentially is you can take real data, and then you use that to simulate realistic data that has those properties, and then you feed that to your models to essentially get them close or in the ballpark of learning this data without going through a whole bunch of data. So you kind of prepare your models and get them in the ballpark. And so if we use this kind of an approach, we actually improve performance as well. You can see here going from about 55 to 60% up to about 70-75%. One type of structure that's used a lot is called a convolutional neural network, and basically what that means is you have you're incorporating imaging information, not just treating things as a sequence. So here we have a patch, we have like a square, those can be cubes as well, and they're slid over your image, essentially something called convolution, and then you just essentially break those down into smaller and smaller blocks, and at the end, if you're trying to solve a classification problem, you might have categories here like dog, cat, boat, or bird. We can embed this convolutional neural network in the context of something called an autoencoder. So what is an autoencoder? That's something where you have your input data over here, and then you have the network gets smaller and smaller, and then in the middle here you might have sort of a code that sort of represents the input images. So maybe it's a classifier, maybe it's just a sequence. And then at the end you have at the output, you sort of expand back out, and then you get a representation of the input. And this is what the model predicts for that output. So we've combined those two concepts of convolution and autoencoding. Deep learning is used a lot on images, obviously with Google and Facebook, et cetera. And brain segmentation is a great example where deep learning does quite well. Here we've trained a sort of dilated convolutional deep network using free surfer decompositions, and we show that we get comparable and in some cases slightly better segmentations when we run it on unseen data. And the nice thing about this is you don't need a ton of data because it's using the samples or the voxels in the image, and so you can train this on 20, 30 images and you can get a reasonable performance. And we can also do this in looking across multiple surface-based ROIs and with multimodal data. And so once you train this model, now you have the model, and then to apply it to new data is relatively quick because your weights are now fixed. And so we can do essentially a free surfer quality decomposition in just a couple minutes. As opposed to on one processor, it can take a half a day to a full day to run the free surfer, native free surfer. Okay, so that's structural MRI. Let's pivot to functional MRI. And so here again is an example of an autoencoder where we're trying to train, say we're trying to learn numbers. And if we iterate the training, you can see that you start with sort of blurry numbers that don't look very well. Close to the original. By about the 9th epoch, we're getting pretty close. So we can use fMRI simply as a, features derived from fMRI. So we can take a connectivity matrix or a functional network connectivity matrix, which is shown here, which we're all probably pretty familiar with. And then you can simply train on those to classify for disease versus non-disease. And here what we're doing is we're using a stacked autoencoder model to do that. I'm not going into the details. You can find the details in the paper there, and hopefully you'll be able to get the references downloaded from the website. But using a deep model, we do substantially better than either a linear or a nonlinear support vector machine. The error drops by close to 10% when we're classifying schizophrenia versus healthy controls. And then, as I mentioned before, we can look at the various layers to see how the connectivity represents at those different layers. And red and blue represent a higher in patients versus in controls. Now, another model that has become quite popular is called generative adversarial networks, and essentially what those are doing is you've got a generator here, and we've created sort of noise data, and we're comparing it with actual data, and then this model here is trying to learn this data and create matrices that might fool the actual classifier that we're interested in here. Here we're looking at major depression versus healthy controls, and then you've got this other arm here that's basically trying to trick it. And it's actually a pretty interesting model, and it can improve performance quite substantially in a number of cases. Here we're comparing across a number of classification approaches. It's not a huge performance, but it did jump up about 5%, going from, here's linear SVM, deep neural net, and then here's the GAN model, separating healthy controls from major depressive individuals. Okay, so that's still using the fMRI connectivity matrices, but we actually have time courses from the fMRI, and so we can use, to do that, we have to incorporate the concept of time into these models, and you do that with something called a recurrent neural network. And so there you have a sequence where you're looking at sort of previous and future. Some examples of recurrent models are gated recurrent units or long short-term memory units. And using that, we can essentially incorporate a memory and try to learn from the time series information from fMRI data. Here's an example of a gated recurrent unit where we're actually giving it time courses that were obtained from group ICA or from Atlas-based measures. And we're training to separate, again, schizophrenia versus healthy controls. And I'll just kind of go through this quickly, but you can see that we again have shown that when we directly compare with other classifiers, we are outperforming, for example, the support vector machine approach. There's a small variation between different types of gated recurrent unit models that we used here. And if we compare the information from the time courses that were extracted from either ICA or a low-dimensional atlas called the AAL atlas or a higher-dimensional one called the Brainatome atlas, you can see that when we're using this sort of data-driven parcellation, we actually do slightly better in terms of our performance as well. Again, we have to go back another step and get at creating atlases using deep models as well. Here's a visualization of the results where we're seeing testing and training for healthy controls and schizophrenia patients presented on a two-dimensional plot. One sort of caveat for these is that this is really a high-dimensional space, and so that two-dimensional plot is going to be lossy, right? We've lost information, so you can't really base everything on the plot that you see here, but it can be useful to see what's going on, sort of at a high level. And then the top discriminative components here were cerebellum and basal ganglia. Okay. And then doing what I mentioned before in the Huntington's data, we can do the same thing by looking at symptoms. We can look at, for example, positive symptoms. Here you can again see the sort of most severe positive symptoms are sort of furthest away on this arm, and then the least severe here are closest to the healthy controls. And that's true for also, total score as well. Okay. We can also do a sort of a dynamic prediction. So instead of focusing on predicting something about an individual compared to another individual or compared to a group, we can look at sort of time points, the sequence information in fMRI. And here we're using, again, a recurrent neural network ICA model. We've been working on trying to blend together deep models with matrix decomposition approaches like ICA because it helps, I find, with the interpretability in that we get this grouping of networks which we're trying to relax with these deep models. And so here what we're doing is we're predicting, you can predict, for example, the occurrence of a response to a task, or you can predict sleep state, for example. We've done that in this paper, looking at predicting auditory oddball responses or predicting sleep staging data coming from EEG that was collected concurrently. And then lastly, I mentioned sort of atlas building. Here, if we use nonlinear ICA instead of linear ICA, we can start to get resting networks that are a little bit more flexible and allow us to capture nonlinear relationships that maybe would have been missed using sort of standard linear models. And this is some work that we've sort of built on and worked together with Aapo Hyvänen, basically doing capturing information using something called spatial contrastive learning to get resting networks in a nonlinear ICA decomposition. Okay, and then the last couple topics is to think about multimodal information. So here's an example where we're looking at brain structure and brain function, which obviously are related to one another, but in a complex way that we don't fully understand. And so what we're doing to try to get at that in this case is to combine something called an attention model, where here you've got a bunch of images and you've got a caption below each image. And if you attend to a particular word in the caption, you want the model to tell you where in the picture is that word relevant. Okay, so here's frisbee, here's girl, and it highlights the face of the girl, for example. And then this is an alignment model. This is like a recurrent model that incorporates time, where then you use these to do things like translate between English and French, and you might have one word go to two words, or you might have the order of the words be different. And so what we're doing in the context of brain imaging is we've got structural features that we've again extracted with ICA. So these are covariating structural networks using what's called source-based morphometry. These are our functional features. We've used a dynamic connectivity approach to essentially get we have this sort of sliding window, and then we've clustered it, and then we've identified states. When in a given subject are they in each of these states? Okay, so this is a state vector, and so the state vector is kind of like the caption in the previous example I showed you, and the structural data is kind of like the picture itself. And so what we've got is a model where we've got this sequence predictor that takes the dynamic functional network connectivity as input, and we've got this attention network that has the structural information that comes from the gray matter. And using this model of alignment, we find substantial alignment between structure and function. So there's obviously a strong relationship between the two, highly significant in terms of alignment. And also there are some significant differences between, for example, patients and controls. So if we just maybe look up here at the top for a second. So there's a strong alignment here. What we see here is that middle temporal gyri is strongly linked or aligned to state 3 in the dynamic functional network connectivity approach. And that alignment also correlates with cognitive score. So there's a lot of interesting features here, and I think we're still unpacking, how to interpret it and what it really means. But I think this kind of work is really important. And if you go also to looking at genetic data, which is even more complicated, but presumably more nonlinear as well. If we just use a simple sort of stacked neural network where we have unimodal sections for single nucleotide polymorphism array data and structural MRI gray matter segmentation data, and then we've got a multimodal layer here that combines the two, and we just look at performance, predictive performance. We see that, and this is pretty common. Structural MRI does not bad. It's in the 70%, give or take, 80%. SNP array data does pretty poorly. Very small effects. You can see a lot of overlap there. But when we combine the two of them together, we actually outperform either one separately. And on top of that, if we use this sort of relevance propagation, or we try to visualize the layers, we can see which brain regions here are most relevant to that, and we also get which SNPs and or biological pathways. In this case, we're looking at, we found the most relevant ones were involved in biogenesis of mitochondria and protein response, autophagy. Okay, and this is just a slide as sort of an extra piece where we are working on some data sharing approaches that are decentralized. And so instead of putting everything into a centralized repository, sometimes data can't be shared for either privacy reasons or other reasons, and you want the data to be analyzed at the separate sites. And so we have a software tool called CoinStack, which enables us to run the data at these different, basically you create a model, and that model gets automatically replicated and runs at each site, and then it gets aggregated centrally at a private aggregator, and then you get a group answer from that without actually ever sharing the data itself. And so within this context, we've actually built some approaches to do neural network modeling, which enables us to do deep learning as well. So this is still very much in the early phases of development. What we've got working right now are kind of the standard models for processing imaging data. We have the algorithms for the neural networks, but we haven't got them fully implemented yet. So if you want to jump in and try it, you won't be able to do that piece just yet, but it's coming. Okay, so in sum, I think deep learning is quite promising for analyzing neuroimaging data. It allows us to learn features that might be more complex from the data itself. We can do things like rapid segmentation, data fusion. There are actually a number of interesting approaches for doing things like image reconstruction and classification that I hope will be discussed today as well. We get good classification performance, although that is not always the case. Again, you should you shouldn't think deep learning is a solution to all your problems, because it has a lot of problems associated with it as well. Difficulties in training, it takes a long time. Sometimes it doesn't work as well as simpler and more easily applied approaches. And much more work is needed to continue to sort of optimize and explore and apply these in a way that sort of can be interpreted and useful for understanding human brain function. There are tons of tools out there for doing deep learning. TensorFlow is a widely used one from Google. So I just kind of list a few of them here. And that's a very non-comprehensive list at this point. We've created a little bit of software in our group. We did some work with a Python-based tool called Cortex, although at this point we're also moving more towards a TensorFlow type framework also. And thanks to the group, to my group, and lots of collaborators that we've been working with to accomplish all of this analysis that I've been showing you. So I think I will stop there, and I don't know if we want to wait for questions at the end or take some questions now. Okay, all right, so does anybody have any questions? And speak loud, or maybe we'll see if the concert hall is good enough for me to hear you. So.
Hi, Very thorough, very clear talk, and to a relative neophyte, overwhelming. It looked like every single topic you described required or was demonstrated using a different kind of deep learning approach. How does one get any perspective on that if one wants to start using these tools?
Yeah, and that was intentional. I was trying to survey different approaches, I think that's kind of what I was getting at before. You can kind of identify main categories of things you want to try to do. So if you want to look at sequences, for example, you need a recurrent model. If you want to incorporate imaging spatial context, you need something like a convolutional neural network model. And if you want to do something like predicting or representing data, like translating one image to another type of image, you want to use something like an autoencoder. Beyond that, there's an infinite number of ways to configure these models, and there are new things coming out all the time. It's really hard to keep up in the field, and so I don't have a good answer on that. I think in many cases there is still, we're still learning what these models can do and how to categorize them and how to characterize them well. So there's actually a bunch of work trying to learn about deep learning models. Like we've been using things like dynamic connectivity to analyze deep learning models as well and sort of understand what they're doing. But I think that's a really good question, and it's I guess a caveat to be careful of is in these days of sort of reproducibility and replicability and the garden of forking paths, we've got like an infinite number of forking paths now that we've got these deep learning models. So you have to be really careful that you're doing things in a way that will replicate and that can be reproduced and generalized. So.
Thank you for the great talk. All these methods basically started out with Google, who has like data sets in the size of million observations. And I was wondering what kind of data set size you would recommend to have any chance to get reasonable results?
Mm-hmm. So it depends on the model you're using. So like I showed the example from image segmentation, you don't need that much data to get a good model fit for a segmentation problem, because it's using 100,000 voxels as opposed to subjects, right, to learn the models. If you're doing something likewise for fMRI, if you incorporate the time series information, you have, if you're using sort of a short TR data set, you have a number of time points now that you can learn from. So it really depends. I don't think there's one answer there. It depends on which kind of model you're using. If you're trying to classify individuals, again, more is better. I would say, you want to be in the hundreds to thousands if you can get there. To get at least something that gives you some confidence that it's actually going to work well on new data. So,
thank you.
Mm-hmm. But you can certainly run them on, 100. Yeah, it'll run, it'll work. I just don't know how. I would put a lot of confidence in there in terms of how it would generalize with that smaller number.
So I'd like some advice on constructing priors. It would seem intuitive to me that if you build priors, you'll improve the performance of your model unless you're building in either explicit or implicit assumptions that degrade the performance of the deep learning model. More specifically, I'm working and building connectomic priors using meta-analytic data. And we've shown that meta-analytic data is a good predictor of per-subject data, but are there things I should watch out for in trying to take these meta-analytic network models and use them as priors for deep learning?
I don't know that there's anything I can think of that would be specific to deep learning. It's more general sort of monitoring the influence that the prior has on the final solution, and is it actually going to allow you to capture things that aren't represented in the prior, right? And so that's kind of standard type stuff. We've also been using things like ICA-based priors of connectivity, you know, to in a sense, enable us, avoid the problem of trying to categorize or characterize everything at the end. But the main limitation there is that if there's something that's not represented at all in your priors, you might miss it. So I think if you can blend those together, where you have maybe you have your things that are sort of well represented across many studies, across many individuals, by all means, I think you should use that information, but maybe also have maybe another arm that captures some things that maybe after you get that information, what's left and what might be new and innovative that isn't well captured in the prior. So,
yeah. I noticed there weren't a lot of p-values and effect sizes. And I kind of encounter this problem of how do you talk to the stats people when you're doing deep learning? And is there an easy answer to that?
well, I think some stats people don't like p-values either. Yeah, certainly. So I actually had quite a few slides with p-values. I didn't highlight them. So I was doing, when I'm doing comparisons of models, right, to other models, we try to do statistical comparisons there, so we actually get some confidence intervals and relationships between them. You get a single accuracy out, but you can run, you know, on multiple subsets of data, and you can do comparisons, and you can get distributions, and I think that's, you know, in the end, when you're trying to make some conclusions about some data, you're going to need some statistical context, right? So just showing a picture or giving an answer as a single number, I don't think will be enough. So I think we still need statisticians. So, yeah. Okay, thank you.
Great, thanks a lot. And I'm going to be presenting from my own laptop, so let's try connecting here. Let's see. Seems promising. Okay, I'll introduce myself in the meanwhile. My name is Ariel Rokem, and I'm a data scientist at the University of Washington eScience Institute. And I'm, together with Andrew Doyle, I'm the co-organizer of the symposium. And one of our goals in this symposium is to give you a lot of sense for what it looks like when you're doing deep learning. And for that, we'd like to show you examples of code. You'll see a variety of, as you've already seen, there are a variety of applications of deep learning, even over the relatively brief few years in which deep learning has been a practical tool for use in neuroimaging research. There have been a variety, there's been a lot of creativity in how to use these methods, and you'll see that through the talks that you'll see throughout the day and have already seen. The goal of my talk is to introduce deep learning really from scratch, from a very simple starting point, so that you can grow to appreciate the complexity as it builds on top of that. And for that, I'd like to show some code, and that's why I need to present from my computer. Let's see if this works now. Yeah? Are you getting my computer? Okay. In the meanwhile, I'll just say that a lot of the presentations and links to computational notebooks are available on a website that we've created for this. You can find that at bit.ly/OHBMDL. That's where a lot of the materials are available already, and links to the notebooks that you'll see throughout the day. So I'll try this one more time. closer to the microphone. Okay, yeah, no problem. Is it possible to get my computer on? It's happening? The link again is bit.ly/OHBMDL. And that includes also the schedule for the lectures today. So we've started already with Vince Calhoun. We will continue with me with an introduction. Following me will be Sage Rutherford, who will be talking about convolutional neural networks and how to use those for brain age analyses. Hannah Spitzer will follow with work on segmentation, image segmentation. Let's see if this. And then one of the interesting applications of deep learning has been rather than pointing it at images, instead to use deep learning as a model of the brain. And Grace Lindsay is one of the people who has really pioneered that, and she'll be speaking after that. We'll go out for lunch and come back at 1 in the afternoon. We'll have several talks in the afternoon. We'll have 2 talks that will focus on generative adversarial networks that you've already heard about. One about synthesizing fMRI and the other about synthesizing translation between different modalities of images. This is from Bliss Chapman and Anders Eklund, respectively. After that, we'll have applications to other less common applications to other imaging modalities, a really interesting application to quantitative susceptibility mapping by Stefan Bollman. And then finally, use of diffusion MRI, deep learning in analysis of diffusion MRI data, segmenting tracts there. Finally, Andrew will wrap up things with a summary talk that will be about looking inside of the black box, and I think my computer is up now. Okay, so here is the website I just read from. As I said before, if you go to this website, you can click on these links that will take you to notebooks that I will show in just a minute. Let me start with a few slides just to introduce the topic really from scratch. So deep learning is a field of machine learning. Machine learning is a field of statistics, a field of computing that focuses on learning from data. And learning from data means that we can extract certain features from the data, or we engineer them, we select certain features of the data, and then we use a set of training data to look at those features and infer something, model parameters that then tell us something about these data, their properties, and for example, the examples that you've seen just before. And then we strictly separate out a separate test data to evaluate the accuracy of these models, and that's something that we really need to do when we're doing deep learning, because these models have a very large number of parameters, a lot of degrees of freedom. Artificial neural networks are a family of machine learning algorithms. They're sort of biologically inspired. This is from the work Vince, in his timeline, he had way over on the left work from the 1960s on so-called perceptrons. Minsky and Papert wrote a book about that, and they have this image that I really like that sort of shows how there are neurons on this side looking at the world, and then it turns into math over here on that side. This is another kind of representation of neural networks. You might think of input units at the very bottom, sensing the input data. For example, this might be pixels or voxels of some imaging data, each one of these input units, and then the information propagates to hidden units in the network through a set of weights. So each hidden unit here sums linearly over its inputs, and then it can transform this data through maybe some nonlinear operations. Those might be nonlinear transformations within this, or pooling across different units. And then this happens again in another set of hidden units, and there may be many, many layers of hidden units, which is why these models are sometimes called deep networks. And then finally, there are output units here. These might be of the kind that say, oh, for example, this was an image of a cat, and or if this one is active, this was an image of a dog, or it might be something more continuous. And learning in networks is done by backpropagating the errors. So we learn by looking at the data and then assessing cost function. For example, did we get how many of the data points that we're currently showing did we get right? If we didn't get a lot right, then we need to somehow tell the network about this. And the way that that happens in neural networks these days is by a process called backpropagation, which we will look at in a little bit of detail in just a few minutes. The idea is you might think there's a daemon inside of this, in this network, and it goes from node to node and from weight to weight and adjusts those in some way. And this diagram, I should say, comes from a wonderful online book by Michael Nielsen that really lays this out both in simple language, but also in mathematical terms. So if you want to learn about neural networks, that book, I recommend that book. And so for the top layer, we look at how would the error change, how much better would we do if we change the activity in each unit just a little bit. And we can do that at the top layer because we have access to the errors of the top layer. If we go further down into the network for layer L-1, we need to ask how much would a change in each weight affect the activity in the following layer, layer L, the layer one above that. And that depends on how active is the node feeding into this particular weight. How would a change in the weight affect the output of the node it feeds into? And then how is the activity of this node affecting the error? And we'll see this in detail in a little bit more fleshed out in code, this algorithm, in just a few minutes. And then the second part of training the network is what's called stochastic gradient descent. In stochastic gradient descent, you choose some random batch from your training data, you calculate the errors from the top of the network back propagating through, and then you adjust the weights by a small amount, and you repeat that until the network converges. This is what it looks like graphically. Imagine that we only had 2 weights in our network. The x-axis is the first weight, and the y-axis is the second weight. And the blue and red here are not known to us in advance, but are the errors of the network. And we start by just initializing the network to some random value of these weights, and then we let the backpropagation process explore this and go down the gradient. Notice that the process of stochastic gradient descent doesn't go smoothly directly to the minimum, and that's because in each iteration through, we're looking at a different set of data, and this data might be noisy, and some of the data tells us to go over here, and some of the data tells us to go over there. But overall, the data pushes us towards this minimum of the error, and eventually it converges and settles down there, and then we might stop the learning. So that's the process of stochastic gradient descent. You heard about convolutional networks. We'll look at a bit of examples on convolutional networks in just a few minutes. It's interesting to note that they're inspired by our visual system. Neural networks in general are inspired by the biology of our brain, and convolutional networks are in particular inspired by the visual system. Here's an image of the physiology of the nervous system in optical imaging recordings, where you can see that each part of the brain represents different orientations. But another way to think about this is that for each orientation, there is a filter that explores the full visual field. So you might think that it's more like this. If you think of this 3x3 array down here as a filter, for example, a filter for horizontally oriented lines, that filter is then passed over the entire image in what is a convolution to extract this green image up here. So the blue is the input image, and the green up here is the extraction of this convolution into what we call a feature map within the neural network. And so here's an example of a convolution of an image. If we take this image of a wall, and we take this filter that has a horizontal line in it, then you see that this prefers certain parts of the image. When we filter this image with this convolutional filter, we get parts of the image are emphasized that contain horizontal lines. And if we take a vertical line instead, you'll see that parts of the image that contain vertical edges are emphasized by this. And this is learned automatically by artificial neural networks. These kinds of filters are learned automatically by the networks. And these networks have been really, really successful in tasks like, for example, classifying different images this is the landmark paper in 2012 that Vince was referring to before that really brought these methods to the fore. So, okay, I'm going to switch over now to a computational notebook that you can run on your laptops as well. If you go to the link that's in our schedule here and click on this, it will take you into this environment called Colab, and hoping that the Wi-Fi works for us here. Maybe I'll stop mirroring so that I can see what I'm doing. All right. So this is called the Jupyter Notebook, and it contains code, it contains some text. Each one of these sections of this notebook is called a cell, and you can execute this cell by typing Shift and Enter. And the first cell here will get some, it'll warn you that the notebook was authored by me and not by Google. I'll tell it to run it anyway, and it'll reset my run times. I will approve that. When running things in this environment, you want to make sure that the runtime type selected by going to runtime and change runtime type, you want to make sure that it's a GPU. Graphical processing units, the things that we use to run graphically demanding computer games, have become really, really useful in neural networks as well, because they can accelerate some of the computations, like convolutions, that we do. And so it's really important when trying to do deep learning and using artificial neural networks in computations to use GPUs, because otherwise things are really, really slow. At the top here, I'm executing some code in Python to import components. I will make this larger so that it's visible. How's that? Can you see that back there? Somebody? Yeah? Okay, good. So I'm importing some modules that I'll be using. And as a first example, let's look at a really, really simple neural network that I've diagrammed up here. It's got two input units, x11 and x12, and two weights leading into an output unit. And so I can write this out. So the way this network works so far, it's very, very simple. So the way it works is that the output is a linear sum of the inputs. X11 times this weight, and x12 times this weight. So the activity in the input units is summed linearly. Implementing that in some Python code, I set the weights to be some values, and then implement this equation in simple arithmetic in Python code, and I can print the output. It's 4 in this case. More generally, the output of any unit is a dot product between its weights and the activations of the units impinging on that unit. So here I'm implementing this as a dot product and doing the same, and the result is the same. A multi-layered network will have a hidden layer like here, right? So this is a very simple multi-layered network. It's got the input layer as before, it's got a hidden layer, and so now every unit in the hidden layer receives weights from each one of the inputs. So you see this one receives through this weight 2,1 and weight 2,2, it receives inputs from x11 and x12, and this unit here receives through weight 1,1 and weight 1,2 inputs from these input layers. And then finally there's one output unit that sums from the hidden layer. So this sequential feed forward with one hidden layer. And so I'm implementing that here. I've initialized the weights, the activities of the inputs. Here I'm initializing the weights from the first layer to the second layer to the first hidden layer, and then I'm implementing the weights from that layer to the output layer. And to do the computation of the output of this network, I need to do 3 sequential dot products. Each line here in this first section is the dot product done by the units in the hidden layer, and then this is the dot product executed by the output, by the one unit in the output layer. And so first, these hidden layer activities are computed, and then these hidden layer activities are then taken as inputs into the output layer, and somehow miraculously this network also has an output of 4. Things get a little bit more interesting when the operations done by each unit in the network are not strictly linear operations. So it's very typical for units within deep learning networks to take the inputs, sum them linearly, and then operate on them through some function. Here it's designated through this through this letter F. So the function is operating on these linear sums. For example, we might have one that was really, really popular early on is this hyperbolic tangent, and that's because it looks like the activation of a neuron in our nervous system. This is the FI curve that you might recognize from basic physiology. But another function that's been used more recently is this one. This is called the rectified linear unit, and you'll see a lot of those today in a lot of the code that we'll be looking at. Rectified linear unit, if the linear sum of the activities going into a unit is less than 0, it sets the activity to 0. And if it's above 0, it takes that and passes that on. That's called a rectified linear unit. It's a very simple form of nonlinearity that can be implemented by neurons in an artificial neural network. Here I'm implementing that by taking the maximum between the dot product, that is the linear sum that goes into the unit, and 0. So if it's less than 0, it gets set to 0. And so when we do that, the output of the network is now different than it was before because each unit is now implementing a nonlinearity. As I said before, networks are trained through gradient descent. We gradually change the values of the weights of these weights. We're learning changes in the weights through observing known sets of input-output relationships. And the gradients are calculated. The gradients to change the weights are calculated through a process of backpropagation. Errors propagated back through the network to calculate a gradient, or you can think of it as a derivative on that error function that I showed you before as a blue and red map, by multiplying the following 3 numbers. The gradient of the loss function with respect to the node a weight feeds into, the value of the node feeding into the weight, and the slope of the activation function of the node it feeds into. So those are the 3 numbers that we need to know, and when we multiply those together, it tells us how much we should change the weight that we're considering at this point. So let's say, for example, that we have the network we had above, and let's assume that instead of 12, we want the desired output for the inputs that we put in to be 10 instead. So in this case, we take the simplest possible error. Our simplest cost function is the absolute difference between the output that we got from the network, x31, the output is the activity in the output unit, and 10, which is our desired output. We calculate the gradient of the activation function, or the derivative of the activation function, which is the slope of the activation function, which for the rectified linear unit is simply is a very simple function. If the number is larger than 0, it returns 1 because the slope of this function up here is 1. And if it's less than 0, we return 0, which is the slope of this rectifying part of the function. So this is the derivative of the slope. And so now we have the 3 numbers that I told you about. The first is the error with regards to the output. The second is the activity of the unit feeding into the weight. And then finally, the derivative of the activation function of the output unit. And so up here are the errors, or the gradients on the weights feeding into the top unit, the output unit. We propagate further by taking now the errors that we had up here and propagating those down through the networks. This is the process of backpropagation. So this is the error 311 that we calculated up here, and that one is multiplied by the activity in the previous units and the units in the input layer, and so on, the derivatives of the activation functions and so on. So we calculate all these errors here in the code. We set a very small learning rate. We're going to slowly go down this gradient, and so for each weight, we update the weight based on the error of that weight, the gradient of that weight, and multiply it by a very small error learning rate that we apply to that. And so here we're calculating the network again, and so now the activity of the network has gone further towards our desired output, which was 10, and then we repeat the process. We do backpropagation again, and so now the output is even closer, it's 10.1, and so on and so forth, until we've settled on the right or close enough to our desired output. So this was a very, very simple network. I'm next going to make things a little bit more complicated because it's not practical for us to write this kind of code every time that we want to do deep learning. Instead, we're going to use a library that implements this. The library that I'm going to show you here and that you'll see in a lot of the presentations later on today is called Keras. It relies on the TensorFlow library that Vince mentioned in his talk, but simplifies the programming interface into simple building blocks that you can use to build the networks that you want for your application. I'm going to make some data. I'm importing some libraries again. Here's some data. Here's some very, very simple data. I'm looking at an image, and it has only 2 pixels. So the first row here are the values of the 2 pixels in the image, grayscale values of the images of the values of the pixels in the first image. This is the second image, the third image, and so on. And notice that I've created training data, and I have training labels also. So here are the labels. The first image is of the second kind. Let's say those are dogs, and the first image is, the second image is of the first kind. Let's say that's a cat, and this is again a dog, and another cat, and another cat, and another cat, and so on. And I'm trying to classify those. Let's see how this data is laid out. You can see that this data, here in yellow you have the dogs, and in blue you have the cats, and on the x-axis you have the values of pixel number 1 in this 2-pixel image, and on the y-axis you have the values of pixel number 2. And you can see that it's linearly separable, but it's intermixed in some way, and that's what our neural network is going to try to disentangle. I'm going to use the sequential kind of model, and in my presentation here for now, at least the models are always going to be sequential, feed forward, and I'm going to import a kind of network model called a dense model, dense network layer, and that means that every unit in the previous layer is connected to every unit in the layer that we're feeding into. So here I'm starting, this is what Keras data looks like. I'm initializing a model object here. I'm adding into the network a densely connected layer with 32 units. I'm telling it about the shape of the input. In this case, it's 2 pixels for the 2 pixels in the image. And then I'm telling it I would like a rectified linear unit, sometimes referred to as ReLU, as my activation for this densely connected layer. Then I'm adding another densely connected layer, and this is the output layer of my network. So this is a very simple network. It has 2 layers in it, and the activation for that is called the softmax, and that's an activation function that helps us determine between different options of the output. I compile the model. I tell it to use categorical cross entropy as the loss. This is a loss function that's used for these kinds of classification problems. And I set some optimizer, and I ask it to tell me about the accuracy as it's running. I should have run this first, and now I'm running that. And that constructs the model. And then using the training data and training labels that I have, I tell it, run this for 5 epochs. Epochs are how many times I run over the entire data. I tell it in each batch, take 100 of my points. This is in the stochastic gradient descent process. Each time I'm going to look at 100 data points and use that to go downhill. And then I'm going to look at every epoch. I'm going to look at 20% of the data that I've set aside, that I will set aside automatically as a validation data set. So that goes through and trains that on, and that went pretty fast because it's a very simple network, and it's not bad. It's doing 85% accuracy in this case. I can turn the model around to predict the outputs for some test data that I set aside in advance, and it can tell me what the prediction looks like. So here's a prediction in a set aside test data, again laid out in this spatial layout. I'm going to skip through this a little bit just because for the sake of time, just to tell you that if the data is not linearly separable, if the data looks like this kind of simple network will just not cut it, and will give you this kind of prediction that is not that great. And for that, we need to add more layers. So here, I'm taking the same model that I created before and adding one additional layer into this, an additional densely connected layer here, and running this fit on that, and now it's doing not too bad in doing this. So adding more layers gives the networks more expressive power. That's part of the reason that these very, very deep networks can do really, really complicated tasks, simply put. Okay, let's look at some brain data. So I've put some data online that you can download, and I'm going to do and now I'm downloading it into this machine, and I'm going to try to do a very simple task. This data is pretty contrived. It's sagittal slices through data through brains, T1 weighted images from the IXI data set, and some of the data is flipped so that it's facing right, and some of it is flipped so that it's turning left. And I want to build a classifier that will tell me for each image, is it facing left or is it facing right? And I'm going to try to train a neural network to discriminate between these two cases. So I'm going to use, again, the sequential model as before, and densely connected layers, and it's going and crunching through the data. It's a little bit more data than before. And you can see that it does really, really badly in this case. Unfortunately, this network, even with its 2 layers, is just not strong enough to do this kind of classification. What I'll add in here to add to this is convolutions. Vince mentioned convolutions in his talk. Convolutions are filtering of the image with a weighting function. I have a simple example here that I'll show you. I'm going to skip over this a little bit, just for the sake of time. But here I walk through a really detailed example of what a convolution is. Let's look at a convolution of one of these brain images. In this case, I'm convolving this with a filter that doesn't do much. It smooths the data a little bit. But if my convolution instead looks like this, it's got a line, a horizontal a row of, it's a 3x3 filter with the first row being all negative 0.5, the central row being 1, 1, 1, and then the third row being all negative 0.5. When I convolve that with the image, what that does is it emphasizes the parts of the image that contain horizontal edges. I can also convolve this image with the same filter turned 90 degrees, which has then the vertical bar, and that then looks like this. It emphasizes the parts of the image that contain vertical bars. Convolutions are useful in neural networks because natural images contain correlations. For the same reason that they're useful in our own visual system, the filters that neural networks will learn will pick up on the correlations, the spatial correlations that exist in images. And those exist in natural images, but they also exist in images of the brain. Things that are further apart in the brain are more different than things that are close by to each other. Using convolutions in neural networks allows us to reduce the number of parameters in the first few layers of the network. So the first few layers of the network gain a lot of expressive power by reducing the parameters for every unit. And that's because if you think about the input of the network, it might be an image with 256 by 256 pixels, and if you had to learn a single weight for each one of them, that would be a lot of weights. But if instead all the unit has to learn is a 3x3 little window, that's just 9 parameters per unit that it has to learn for these relatively large input images. And that is how these convolutions save us parameters, at least in the first input layers of the networks. So here's how we express convolutional networks in Keras. We import this conv2D layer from the library of layers that exists in Keras, and then instead of densely connected units here, I use this conv2D. Again, I have 32 units of this kind, so each one of these units will learn a different set of 3x3 parameters for the convolution. I set the kernel size to 3 so that I have 3x3, and then I tell about the input shape and so on. I set the activation to ReLU, and then to pass that on to a densely connected output layer, I first have to flatten the results out between the convolution and the densely connected layer that follows. I can then do the training. This takes a little bit longer here because the data is a little bit bigger, and it crunches through. And I'll just cut to the chase that this doesn't really work that well either, and that's because this network is just not deep enough. So the following cell here will contain the implementation of a network that is deep enough for this problem. It's got 1, 2, 3, 4, 5, 6 convolutional layers, and in between convolutional layers, in addition to the activation, to the nonlinear activation function, the ReLU, there is also a max pooling operation that happens, and that's spatial pooling. The pooling operation takes the images and downsamples them by taking the maximal pixel in every little neighborhood of pixels. And I don't have much time, so I won't really get to the end of fitting this probably, but I will tell you that this one does this task rather well. So what I'll do now is I'll encourage you to go back and look at this notebook. As I told you, if you go to our website, the notebooks are linked from the schedule, and then there are the slides that I showed you before, and oh, here it is, it's done. Because it's running on a GPU, it runs pretty fast, and it's got 90% in this task. So to summarize, the building blocks that we use here are densely connected layers and convolutional layers. We stack those on top of each other into these sequential model objects. We use these sequential model objects fit method on training data with appropriate labels to do the training, and then we evaluate on separated test data and test label to tell us how well the network is doing after it's been trained. And so you'll see repetition of this pattern a lot in a lot of the talks that you'll see throughout the day when presenters are showing their own code. And before we move on to the next presentation, I'll be happy to maybe take a couple of quick questions if you have. Okay, if there are no further questions, then I will present the next speaker for this morning. Thank you. The next speaker for this morning is Sage Rutherford. She is a researcher at the University of Michigan, the Department of Psychiatry there. And she's done a lot of interesting work using neural networks, both in complicated tasks like segmenting images from fetal MRI, and also in predicting brain age, and that's the work that she will be talking about today.
Okay, hi. I'm super excited to be here this morning today in this awesome session, in this beautiful auditorium. Wow moment for me. Thank you, Ariel and Andrew, for organizing this course. That was a really great start to some theory and stuff, and now I'm going to kind of dive into some examples. So my name is Sage Rutherford. I'm a researcher in psychiatry at the University of Michigan, where I've been for a couple of years. And today I'm going to be talking about...
You have to go at the podium because to go on with the presentation, you have to go at the podium.
Yeah, because I need to switch in between code and the talk, so... Let's see if I can stop mirroring. So today I'm going to go over this model that I've been using for brain age prediction, but I also kind of want this to be generalizable in that maybe you use different types of brain images. So in my examples, I'm going to be using structural T1 weighted images to predict age, but maybe you're interested in using a different type of brain image, like a different anatomical scan, or maybe a mean task activation map or something to predict a different phenotype. So as we're walking through this together, maybe keep in mind, ideas you have for how this framework could be applied to something other than just predicting brain age. Because that's, I've sort of validated that here, but I really believe that this model could work well for other phenotypes as well. So I'm going to talk about how we've adapted CNNs to be applied to brain images, some of the modifications we made from these kind of typical architectures, and talk about how I've been working on that the past few years. And also at the end, if we have time, I'll get into some ideas for even further modifications that we could make. So just to introduce myself, this is my Twitter because that's how we know each other nowadays. So I'm an analyst in psychiatry. I've been working there for a couple of years now. I work on a lot of internal kind of clinical data sets, but then also I get to work with really large-scale open data sets like the Human Connectome Project, UK Biobank, the kind of data sets that allow us to train these deep learning models. I'm very passionate about yoga and cycling and dogs. These are my dogs, Charlie and Blue. and my background is in biomedical physics. So this whole project of brain age prediction got started about 1 year ago at the Machine Learning for Healthcare conference in 2018, and there's a link to the paper here. It was led by Pascal Sturmfeld, so I want to make sure to give him credit for the original idea. And so where he started was, he's a computer scientist with no background in neuroscience, and he was kind of starting to work with my group and look at how the neuroscience field was using CNNs and kind of recognized, hey, like, nobody's really making any like modifications for this new application. They're kind of taking these out-of-the-box models and just applying them to brain data without really making any challenges on some of the assumptions that the models make. So in this paper, he kind of started to explore that. And that was also in collaboration with Mike Engstad in my group, Chandra Sripada, who's the main PI that I work with, Mark Petersburg, and Jenna Wiens in electrical engineering. So the first assumption that we kind of wanted to challenge was this idea of spatial invariance. So CNNs were really designed to work as image recognition tasks in images. So say you have a model that is looking to identify a dog in the image. And in that image, in this natural image, I'll call it, it's going to look for a dog anywhere in the image, whether it's here, different sizes, maybe there's multiple dogs. The idea is that it's going to kind of share the parameters across this whole image, and that's why things like data augmentation works really well. You can translate your objects across the image, and the whole point is that you're looking for the same thing no matter where it appears in the images. Now, in brain data, that idea doesn't really hold because most of the data that we're working with has been aligned to group template space. And a different location in the brain represents very different information. If somebody's cerebellum is appearing flipped upside down or in a different location, that probably represents that something is wrong with your data. So our data is all in the different, in the same space, and we want to learn different information from different parts of the images. So we kind of needed to challenge that idea. And so again, the goal of kind of the standard CNNs was to detect an object in the image, but in our case, the goal is to predict brain age. So it's not to detect an object, but to learn features about the image that are going to help us predict age. So we're looking to learn unique information from each location in the image. So the second modification that we made, I'm going to call regional segmentation, and I'll walk you through this figure. I think it'll make a little bit more sense on the next slide as well. So we start with an initial 3D brain volume, and we are using 3D convolutions in my example. Many networks will use 2D convolutions, and what that does is treat each, it only considers a single slice at a time instead of considering the whole 3D shape. So using 3D convolutions was another important modification that we did. So we start with this 3D brain volume, and we're going to segment it into 2 planes as our input. So when we make 2 splits in a 3D volume, we end up with 8 different regions, which are represented in these different colors here. So when you do 2 splits, that essentially create 8 new little input regions. Whereas a baseline doesn't do that split and shares the whole filters across the entire image. Now when you do the splits and you separate the brain into these different regions and channels, an important thing to keep in mind is that you want to have minimal overlap between these regions. So you've done the effort to separate the brain into these different regions so that you can learn unique information about that region. But if you force them to be overlapped, it kind of takes away from that. So just wanted to point that out. So this is kind of a brain example of that last figure. So we start with our 3D brain volume. We apply 2 segmentations, and that's going to get us to these 8 different regions that we'll input into our network. And I just wanted to make note that these well, they're not represented here in the figure, they're still in 3D. It's really hard to make 3D figures. And they also need to be the same dimension. So even though there might be a different amount of brain in each channel, like the actual physical size of them need to be the same because the network needs images of the same size, otherwise it gets a little bit more complicated. So the third kind of challenge that we made to the typical CNN architecture was to apply more filters early in the network. Typically we'll see you'll start with a few filters right at the beginning, and later in the network you'll have many. As you get deeper into the network, you'll have many filters. Like here in this example, they start with 64, and their final layer has 512 filters. And when you're working with a model that has very distinct classes, so go back to like the animal example, if you have a model that's looking to maybe classify different types of animals or something, the filters very early in the network might be shared across your classes. So if you're classifying animals, you might have, one of your filters might be like a circle because all animals have a head. But as you get deeper into the network, your filters are really more specific to each of your classes. But when our classes are age, there's not such unique information about the different classes that you're classifying. So we kind of just had this idea like, hey, like let's flip the filters to the beginning. And that actually seemed to work really well. So this is the architecture that we're going to go over today. The baseline approach is is not doing any of the segmentation. We have 4 convolutional blocks followed by a hidden layer in our output of the model. So we take in a 3D image. The output is just a single number, which will be our age prediction, but it could eventually be essentially any number. It could be a fluid intelligence prediction or something. So the baseline is what we're going to compare to show that our modifications are actually improving the model. So in our proposed approach, we do the segmentation, and we do this reverse layout where we start with 64 filters, and our final layer only has 8 filters. And this is just a representation of what a convolutional block looks like in our network. Thought I'd throw that in there. So I wanted to, before we get started with the code, kind of demonstrate a couple of data sets that this model has worked super well in. So in Pascal's original paper, he worked with the Philadelphia neurodevelopmental cohort data set. We had 724 subjects, and the age range was 8 to 21, so developmental sample. And he showed that our proposed approach, where we do the reverse filter layout of 64 filters at the beginning, 8 in the final convolutional layer, and 2 segmentations, we had a mean squared error of 3 years, a mean absolute error of 1.4, and the entire network trained in just 40 minutes. We tried a couple of other experiments. We tried just doing the segmentation only, so splitting it into those regions and not trying the reverse layer, and that was, it trained a little bit quicker, but it was very similar performance. In another experiment, I've tested this idea on the UK Biobank data, which was a much larger data set, so we went from 700 subjects up to 9,000 with a much wider age range, and impressively, our mean absolute error was only 3.2 years, and while the network took 6 hours to train instead of minutes, it was a lot more data. So this is actually still quite an impressive result that I was excited to see. So I wanted to point this out. I mentioned, doing this segmentation really helped the network, but we found that there is kind of a limit to this. The more you start to segment, you'll actually start to do worse, and we think maybe that's because you're just sort of giving too much information, or there's maybe not unique information in all the different segmentations you're doing. So that is just something to keep in mind. So, okay, a couple of details about today's code. So while I just presented data from the UK Biobank and PNC data sets, Both of those data sets are not the easiest to access. Like UK Biobank took us quite a long time and a bit of money to be able to download and access. And both of them have kind of a little bit stricter data use agreements. So I wanted to use data today from the Child Mind Institute because A, it's a really great developmental data set that I think more people should know about. But also the data use it's much easier to gain access to that data. So if this is something that you want to, play around with a bit more when you get home, this data is really easy to access. And they have structural data, there's functional data, DTI. It's a very rich data source, and there's also a lot of phenotypic data if you're interested in applying this, like I said, to something other than age prediction. And so I also, I introduced this idea of just making this random split in the brain, just right down the middle twice. Today I actually want to introduce kind of a more anatomically defined split. Like we know that, there's maybe unique, there's better splits and segmentations you can do of the brain other than just, splitting it in half. So today we're actually going to look at splitting a T1 into gray matter, white matter, and CSF masks and look at how that helps this model. And also just here's a link to the data portal for CMI, and there's a wonderful scientific data paper as well. So with that, we'll get started with my Jupyter, or with my Google Colab notebook. Okay, I'm connected. Oops. Maybe if I end this. Okay. The display. Just trying to get it so that you can see my code. Nope, still can't. Let's see. Show mirroring options. Arrangement. Somebody want to come help me quickly? Sorry. We'll get this started. I have my displays up.
Arrangement, displays, arrangement, and then check that mirror displays.
Oh, small little box. Okay, cool. Okay, perfect. So like Ariel said, this notebook link is on the Brain, the website next to my name.
Could you increase the size of the font, Command plus?
Yeah. Is that a little better? Okay, we might get a bit of text cropped off, but I think that should be okay. So I, again, have a link. All of the code that we're going to be running today is in this notebook that we're going to run through together, but there's a couple of steps that I had to take in preparing this data. So if you're interested in looking at that, I do have a link to a GitHub here where I've linked some of those scripts because while running deep learning models seems really easy when walking through a simple notebook like this, that's often not the case, and we spend a lot of time preparing our data, or what I like to call data engineering, doing things like making sure all of your input images are the same size, quality checking, silly things like, your files need to be consistently named in a machine-readable thing, so sometimes you might need to resample or crop your images, so I've linked a few of those scripts in my GitHub if you're interested in looking at them. So let's get started. We're going to, we'll author by Google, run anyways. Hope this works. Initializing. So I'm going to install the requirements that I need for this package. They're probably pretty similar to what Ariel just used, but I think I have a few extra. So import those. Guess there's a lot of them. I forgot to do this poll right at the beginning. Can I get a show of hands of who has ever used a CNN before in their research? Okay, cool. I've used a couple of them, but maybe about 1 year ago, I presented at ISMRM a convolutional neural network, and it was my very first time. So I was just reflecting on how far I've come in a year. It's running a little bit slowly today. Okay. So I imported our packages. I'm going to use Keras, which was the same library that Ariel just walked through in his tutorial. I import a couple of neuroimaging packages here, like NiLearn, which is a really awesome library that you should check out if you're not familiar with it. It has some really great image processing tools, like cropping the images. Okay, I'm going to download my phenotypic file to get the ages and the subject IDs. This is also the file that I used for splitting my data into train and test, which I've already done because that took a little while. So I'm going to read in my ages, look at the distribution. So we have 906 subjects. The mean age is 10.8. Look at a histogram of them. So a little bit more skewed toward the younger folks, but it does go up to about 21 years old. I'm going to download the training and test data really quick, unzip it. Oh yay, this is going much quicker. Make sure it's all there, yep. Okay, so I mentioned I had already sort of converted this into our train test split. I use scikit-learn to do that, and I've already put the data into train and test NumPy arrays, which is how Keras likes to read it. Not, but like I mentioned in that GitHub repo, I have scripts that will take nifty images and convert them into this exact format of NumPy arrays if you're interested. So get my labels. I'm going to check the input shape. So the first shape is our batch, or our number of subjects that are in the training. Then we will look at essentially the XYZ dimensions, and then the fourth channel is the number of segmentations. So we're using white matter, gray matter, and CSF, so we have 3 channels there. And this is important because you need to specify in your model when you're building it, as I'll show you down here, you need to specify this variable input shape to the very first layer. The other layers will kind of learn from this first layer, but this is a very important thing to specify. So if you were using this with a different size data, you would need to change this to match your data. So won't go over all these layers too much because it was kind of what I showed you in the slides of 3D convolutions, max pooling, and I do this model summary, which shows us essentially all the information about the model that I just created, the number of parameters there. It's kind of a cool, useful thing. So I'm going to save out some of the checkpoints as we're training this, just a bunch of code to do that. I've set the validation loss here. I've set early stopping. So what this means is if our network is not getting any better in the validation set, which I don't think I defined that. So we have our train set, our validation set, and our test set. The test set is held out and not used whatsoever during training, so it's a truly out-of-sample test set. The validation set is kind of a test set, but it's used, the model looks at it when it's training, and it's kind of tells the model if you're improving, so through every number of steps, it makes a prediction in the test or in the validation set, And this early stopping value here says that, hey, if in this many of iterations through, if we're not getting better in our validation set, or maybe we're even getting worse because we've overfit our model, this says stop. I think here I've set it to 10 epochs. So if after 10 runs through all of our data, if we haven't gotten any better performance, you might as well stop because you're wasting computing power, and you might actually be hurting your performance. Some of these values here are kind of just saving some of like our outputs. Did I run that cell? No. So then I'm going to compile my model. I'm using mean squared error as the loss function. I'm also saving out mean absolute error. If you were doing something like a classification, maybe you would also want to add in accuracy here, but because this is more of a regression, a continuous thing, age here, I'm not going to use that. So I compile it, and the next step is to actually train. So this cell, I had to use this when I was training on my server because we have multiple GPUs, but it's just a little line of code that will show what GPU you're using. Okay, so here I've set the number of epochs and the batch size. This is maybe a little bit not standard here, but for this example where I'm kind of trying to train the network quickly, I've set my number of epochs to 10 and the batch size of 20. And also I set my validation split here. So what that says is I'm going to use 10% of my training data as my validation test set. So we'll set those parameters. And that is going to run, I think it took about 8 minutes. So I'm actually going to hop back to a couple of slides while this is going, but I'm just going to watch for a moment to make sure that it's getting started and how it's going. Okay, it looks like it's running. See if I can not. Already showed you that. So I just wanted to chat a little bit about some different ideas I had for this. So today I was showing segmenting. I showed you this idea of regional segmentation where we just kind of do this random split down the middle of the brain, and in our code we're doing a split where we use white matter, gray matter, CSF, but as we know in the field there are numerous different parcellations and atlases. So I've really been thinking about how I think this architecture could be sort of a way of testing those, you know, maybe for different data types, different parcellations or segmentations would work better. But also keep in mind the figure I showed you where the more segmentations we do, sometimes we can actually start to do worse. So maybe a functional atlas that has like 260 regions or something might not be the best here. Maybe we're better to stick with anatomical atlases that have, I think this one here that I'm showing has like 7 different regions. But I think that this model could be a way of sort of testing out which ones work for different phenotype predictions. I also mentioned like the idea of lesion analyses here. Maybe if you do want to use a functional atlas that has something like 200 ROIs, well, you could test just using a portion of them and then looking at how your prediction performance changes. So I think that could also be interesting. And again, I mentioned that there could be different data input types. So this model is currently set up to work with a 3D volume, so it can't handle like a functional time series, but it could handle something like a mean functional resting state image or a subject's task activation map or some kind of DTI summary image. So I think all of these would be really cool ideas to try out. So I wanted to point out this paper. So we kind of, this is a paper called EfficientNet, and I'll explain it a little bit. So we've kind of been talking about like all these different parameters you can tweak, different inputs that you can have to your network, different atlases, different number of filters, different how deep your network is. And these are a lot of parameters to just start trying at tuning yourself. And this is a really exciting paper that's actually being presented I think the conference is going on right now at the International Conference on Machine Learning, and I think the computer science field was super excited about this paper. And the idea seems really straightforward to me. I was kind of surprised, like, how did nobody think of this sooner? So the idea is that you train an initial neural network with, sort of a very simple one. Maybe you wouldn't do the regional segmentations. It might just have a couple layers deep, a very simple version, but you're inputting, your image and you're outputting your phenotype. What this kind of network introduces then is it tunes its hyperparameters itself. So what that means is it essentially chooses how many layers you should have, how many filters you should have at each layer, how many segmentations you do. So this idea kind of takes that, let's just try out a bunch of stuff and see how it works. So I think that this network could be really useful for this model that I'm introducing you today, because it can be very computationally expensive to train, so many different networks yourself. I think just in preparing for this talk, I trained like 10 different versions of this, and while I have my own server and GPU access, like it can get a little out of control sometimes. So I just wanted to point this out, and I do have a link here to the paper and the code as well, and so happy to discuss this further. I have yet to actually try it out myself, but I was reading the paper and just thought, like, wow, this is so cool, and I think it would be really applicable to this kind of architecture. Okay, another thing that I wanted to point out was this issue. So when we're predicting age, obviously the kind of thing that we're optimizing for is to say we want to get really good at predicting age, and that's the only thing that the model is caring about. Let me minimize the mean squared error between what I'm predicting and what is the true age. So we end up kind of having this forced correlation. And I guess that's not really a problem if your only goal is to predict age, but typically, at least in our lab, we have kind of like a further thing we want to do. In our case, we're looking at, after we get these predictions for age, is there a deviation analysis? So for people who the model is predicting that they look younger or older, or something, there's these extreme deviations in their age predictions. Is there anything, interesting about them that we can learn? I think originally we wanted to show that people with younger looking brains exercise more, which sadly was not the case, at least in the UK Biobank data. But so when there's this really strong correlation between predicted and true ages, we can't do as interesting of a deviation score analysis. So I haven't totally solved this issue yet. Something I've been doing in post-processing is to sort of force our sample into these like quantiles or deciles to sort of force there to be no correlation between predicted or deviation score and age, but I think that's not an ideal solution, and I'm kind of actively thinking about how we could include this in our model. I was very excited to see that there's actually a machine learning challenge this year. I think it's related to OHBM. But it was to using structural images predict age, and I was excited to see that they're thinking about this too, and that they'll give an award if your model also has a Spearman's row of like less than 0.1. You'll have the bias reduction award. So this is definitely something that I'm actively thinking about, and I think is important to address. And then I just linked some additional resources that I've been using myself. I know that there's kind of a overwhelming amount of information out there on the internet in general, but especially with like deep learning, if you're not familiar with it, you're in neuroscience, not computer science. I actively follow the literature and even find it super hard to like keep up to date on everything. So for me, I just decided I was just going to start looking at a bunch of people's code, and I didn't set out to be like, I'm going to learn deep learning and just master it. Like I just started looking at a few examples where people were using it in their research and just following along, even if I didn't fully understand everything that they were doing. I really have found the most helpful thing just to start reading people's code and seeing how other people were building their models and such. So I just thought I would point that out. Okay, now let's go back to our model and hopefully it has finished training. Okay, so we got through 10 epics. Was that how much? Okay, it did finish. So let's look at our performance. So after we fit the model, the next step naturally is to test it on our test set. I think our test set in this case was like 20% of the data, so it's maybe like 160 or so subjects, 182. Okay, and so this time through our these two values, the first one, 28, is our mean squared error. The second value is the mean absolute error. So this actually seems to do not that great in this case. I mentioned in the other data sets our mean error was around like 2 years. One reason might be that we didn't get through very many epics. I think in the other data sets I got through at least like 30 or 40 before it stopped training. Also, I'll mention that the CMI data set is multi-site, which can be an issue. Maybe there's some scanner invariances and there wasn't like balances across the training and test set. I don't think I considered that when doing the split, so that could be a reason why. But so I think that trained in about like, I don't know, 8 minutes or so, which was pretty fast and cool. But if you are running this yourself, I would recommend increasing this number of epics that you're getting through, and I think that would increase your performance. So I just want to go through how to save out the model, because that's an important step after you've spent the resources to train this model. You don't want to just lose it. You want to save it and maybe be able to use it later. So this is a line that will just save it out. It will save, I've sort of listed, it saves the whole architecture of the model. It saves the actual weights. It saves the state of the optimizer. And I think I had a line up here that says, let's find it. So I'm saving like the best weights of the model. It essentially saves like when the model stopped doing the best. So even though we might have gone like a few more epics and where we stopped increasing on our validation set, the model will save wherever it did the best. So that's important. But you do need to specify that when you're designing the model. And then also I wanted to show you guys how you can load in a pre-trained model. I've shared a couple of models that I saved when I was doing this. So we need to import another library. I saved a couple of other models where I had different batch and epic sizes. I think this one, and maybe we can see how we perform on those. And if we do any better than 4 years. So load those guys in. So one of them I had batch size 10, epics 20. One was batch size 5, 30 epics. And here these go through the summary, but it's the same exact model, so I don't feel the need to print it again. You do, once you load in a saved model, you need to compile it again. So this is a kind of a repeat of the line that we had above from right before we trained. But then we're just going to use the same evaluation function, and it is the same test set that we just test the model that we trained together. So, okay, so this mean absolute error is 2.9 years, or almost 3 years, but a little bit better. I think the other one was 4.9. And this was 3.1. So increasing the number of times you go over the data set increases your performance as expected. Okay, and I already finished my slides. So with that, I just want to say thank you again to everybody, to the organizers, and I'm happy to take any questions that you guys have about what we just ran through.
Thanks, Edge. I've got multimodal data, I've got MRI and PET, and I'd like to use them together. And it seems that's something similar to what you did with the grey matter, white matter CSF. Just so I got it clear, did you just combine them into a single 4D image? Cool.
Yeah, as long as they are they're the same image dimensions in 3D, this architecture could handle that. And I would be curious to see how different modalities performed.
And then if I have, if I want to enter other information that isn't an image, so in my case it would be whether it's a left or right epilepsy, could you put that into your convolutional neural networks?
Yeah, do you mean like, kind of the idea of regional segmentation where we just did that split and you would want to have the right and left like as separate, or is it not an image information?
It's not image information.
That definitely can be done, not in the current model as we just walked through. Like, are you trying to predict left or right, or?
No, I'm trying to predict a surgical outcome.
Okay.
And left or right is a predictor variable, but not imaging data.
Yeah, I mean, you could test like just maybe only giving the network the left hemisphere at a time and seeing how that works, and have a separate network to only use the right, and then see how the classification, maybe you can classify only using the left hemisphere, or maybe the right hemisphere does, but it would sort of take like doing a segmentation and then testing only giving the network the left information and then only giving it the right.
Okay.
Does that make sense? I'm happy to talk more offline if you have
that'd be awesome.
question.
Thank you.
You're welcome.
Okay, for such a question as estimating age from image data, you worked with MNI normalized data, but I imagine there's a lot of information also in the non-normalized data. Would it make sense to include them in such a model?
Absolutely. I think that even not normalized data, the data is still in like close enough space that the idea that I brought up of spatial invariance still really holds. And as long as all of your images going into the network, even if they're not like aligned to template space, as long as they were the same size again, that could definitely still work. Or even like free surfer data, a different template or something could definitely be used here.
Thanks.
Thank you for that talk. I recently began using machine learning in the similar context of predicting brain age. And using simpler models such as penalized regression, what I find is some of the top predictors are actually quality control metrics, even in data where you exclude some of the worst scans. So I wonder, I don't know much about deep learning, but how can you know if that is the case with deep learning models, and what can you do about that?
So I think it's definitely important to know your data going in, and one of the negatives about these models, or one of the criticisms, is often that we don't know maybe what information they're using to make their prediction. So like it could be some kind of noise feature or something. So I think it's very important to, there's still not a replacement for visually looking at your data going in. And if you have poor quality data that's going into the model, that's not going to help you at all, and it's not going to help you learn anything new about the brain. So I still highly recommend that you look at all of your data before and make sure that you have high quality train and test, because that could also, like if your data in your training set is all super high quality, and then somehow in your testing set there's low quality data, that would you wouldn't do very well in the test set. So.
Thank you for the talk, and I think I just missed your reasoning behind the segmentation, what you talked about first, so could you just really shortly reiterate on it?
So with segmenting, if you don't do that segmentation, the network will share the same filters across the entire image. And so the segmentation forces the network to learn more specific information for each region, because we believe, like in the example I did with the dog, we're trying to essentially find the same object across the entire image, whereas in the brain we know there's really unique information in each region, and so we want to kind of force the network to focus on that.
Thank you.
Yeah.
Great, okay.
Thanks again to Sage. I'm just here to tell you that there's now a break until 20 past the hour.